Welcome back to Dr. Kokoza's biology class. This unit will be on meiosis, or the division of sex cells. As we learned previously, body cells divide by mitosis. In eukaryotic organisms, mitosis doesn't work for all cells, and we're about to learn why. In organisms that reproduce sexually, which things like humans and cats and dogs and mice do, we need to be able to reproduce sex cells having half the genetic information coming from the parent that is male and half the genetic information coming from the parent that is female. Males donate a gamete called sperm, and females donate a gamete called eggs or ovum. A gamete is going to end up with half the chromosome numbers of a somatic or body cell. Remember that in humans, this number is going to be 23. Let's learn some vocabulary. Diploid. A diploid number refers to a cell which carries a double set of chromosomes. This would be somatic or body cells. So in other words, in humans, we have 23 pairs of chromosomes. Haploid. This is a cell that has half the number of chromosomes that a diploid cell would have. This is going to be your gametes. Again, a gamete is a sperm or an egg. In humans, it would mean that it has 23 total chromosomes, not 23 chromosome pairs. Homologous chromosomes. A homologous chromosome refers to two chromosomes which are similar in structure. Each contains the same genes. So in other words, when we looked at karyotypes, it would be both of the chromosomes in the number 1 slot, or both of the chromosomes in the number 12 slot. Meiosis, what is it? Meiosis is the production of gamete or haploid cells. At the end of meiosis, we will not end up with 2 cells with 100% genetic information. We'll end up with 4 cells that have half of the genetic information, and each of these cells will not be identical. Another way that you can say this is that we'll end up with 4 haploid daughter cells. The original diploid cell is going to end up going through 2 divisions. The first one will look very much like mitosis, and then the second one will end up with the 4 haploid cells because it will not go through interphase. Here is a picture of the stages of mitosis. You can see here on the left-hand side, interphase, then prophase, metaphase, anaphase. Telophase and cytokinesis are left out, but they do occur. Now you can see that you have 2 cells. These 2 cells are then going to divide again. They're going to go back through prophase, metaphase, anaphase, telophase, cytokinesis, and end up with 4 cells with genetically different information. During meiosis, pairs of homologous sister chromatids pair up into something called a tetrad. So in other words, all of the genetic information under slot 1 in a karyotype would be a tetrad. So would everything under slot 2, slot 3, all the way down through the chromosome slots, because they are pairs of chromosome under each number. When sister chromatids are connected in a tetrad, they will exchange genetic information in a process called crossing over. Crossing over is going to become really important down the end of the year when we discuss evolution, because this is one of the ways that organisms are not genetically identical to the parent or to either parent, but genetically similar. Again, this is why you look similar to your parents, but not identical to your parents, because when the sperm of your father met the egg of your mother, crossing over had already occurred, and only some of the genetic information from either parent is going to end up connecting to form the organism that is ultimately you. Again, this is extremely important for genetic variation. And a good way to imagine it is that if you had a brand new deck of cards and you shuffled the deck of cards, it would no longer be in the same order, but it would still be the same deck of cards. Thus, the total genetic information is getting passed on from your parents, but it just may not be in exactly the same order. Thus, you look different, though similar to your parents. Here is a picture of crossing over. You can see that we have the orange chromosome and the maroon chromosome, and that during crossing over, a portion of the orange chromosome, little a, is going to get cut onto the maroon chromosome, big A. So the total amount of genetic information is conserved. It's just that now a portion of the maroon big A resides on the orange little a chromosome, and a portion of the orange little a now resides on the mauve big A chromosome. Again, during meiosis 1, a diploid cell, 46X, goes through a division just like mitosis, producing 2 diploid daughter cells with 32X each, or 32 chromosomes each. After the first division, 2 daughter cells, each with 23 chromosomes, go through a second division without going through interphase. When this occurs, or the reason that this occurs, excuse me, is that because when they don't go through interphase, they also don't go through S phase, and it's during S phase when the DNA is replicated. After the second division, there will be 4 daughter cells that now have half the original DNA of the parent cell. In other words, having 23 single or 23 haploid chromosomes, unlike the division above in which there were 23 pairs of chromosomes, or the diploid number. If you can see the screen now, it says mitosis versus meiosis and a karyotype review. You're going to find a second video that will cover all of this information and start on this slide.